नानक नाम दा ताज इलाही सीस सजावण वाला, जिस दी होंद च शब्द गुरु दा जगे पुनीत उजाला। सदा सदा पुरनूर रहे, सदा सदा पुरनूर रहे। गुरु नानक देव विश्वविद्याला, गुरु नानक देव विश्वविद्याला। सदा सदा पुरनूर रहे, पुरनूर रहे, पुरनूर रहे। गुरु नानक देव विश्वविद्याला। अनहत नाद बजे, आहत जलन बुझे, अमृत शब्द वरे, तन मन तीन हरे। सदा सदा पुरनूर रहे, पुरनूर रहे, पुरनूर रहे। गुरु नानक देव विश्वविद्याला। धरती अंबर पवन समुंदर सूरज चंद सितारे, एक दी तुन विच गावन वावन राग इलाही सारे। सत सुहान सदा मन चाओ, सत सुहान सदा मन चाओ। एह गुरु वाक उजाला, सदा रहे परगास रूप। गुरु नानक देव विश्वविद्याला, गुरु नानक देव विश्वविद्याला। चिंतक सृजक रसक विनोदी, सहज निमर विसमादी। निर्मल निरभव सच दा खोजी, विगसे आद जुगादी। रोशन हो वंडे रुशनाई, रोशन हो वंडे रुशनाई। दिखया पावन वाला, गुरु नानक देव विश्वविद्याला, गुरु नानक देव विश्वविद्याला। जिथे मिलदी ओट ने ओटियां, पौंदे मान ने माणे। ओह दर उसदा, ओह कर उसदा, एह गुरवाक वखाणे। जिथे मिलदी ओट ने ओटियां, पौंदे मान ने माणे। ओह दर उसदा, ओह कर उसदा, एह गुरवाक वखाणे। ओथे नदर मेहर दी होवे, ओथे नदर मेहर दी होवे, जिथे नीच समाला। गुरु नानक देव विश्वविद्याला, गुरु नानक देव विश्वविद्याला, गुरु नानक देव विश्वविद्याला, गुरु नानक देव विश्वविद्याला। नानक नाम दा ताज इलाही सीस सजावण वाला, जिस दी होंद च शब्द गुरु दा जगे पुनीत उजाला। सदा सदा पुरनूर रहे, सदा सदा पुरनूर रहे। गुरु नानक देव विश्वविद्याला, गुरु नानक देव विश्वविद्यालय सदा सदा पुरनूर रहे पुरनूर रहे पुरनूर रहे गुरु नानक देव विश्वविद्यालय अनहत नाद बजे आहत जलन बुझे अमृत शब्द वरे तन मन थीन हरे सदा सदा पुरनूर रहे पुरनूर रहे पुरनूर रहे गुरु नानक देव विश्वविद्यालय धरती अंबर पवन समुंदर सूरज चंद सितारे एक दी तुन विच गावन वावन राग इलाही सारे
Hi Uday.
Hello.
Hi. Yeah, I just wanted to check if your audios are working. Yeah. Hi. How are you?
Yeah, good. How are you doing?
Nice to see you after a long time.
That's right.
Yeah. So everything is working fine. Do you want to check your screen share as well?
No, it should work.
All right.
It should work. When you give me the green signal, I will start.
All right. So we'll start in another 2 minutes. I think. Vijay, is that okay? You have to unmute yourself.
Yes, yeah, I did that. Yeah.
Hi, Vijay.
Yeah, please go. Hello, sir. How are you?
I'm good. How are you?
Very good. Thank you. Nice to have you here, this platform.
Nice to see all of you.
Oh, yeah, thanks.
So we can wait another 2 minutes. I mean, we can start at exactly 4.
Yeah.
Yeah. All right. Great. Yeah. Hi. Hi, Vijay. I think we can start maybe.
Yeah, hello.
Hi, Yatin. Yeah.
Yeah, so I was just waiting for Vandana ma'am, if she is here.
Yeah, I am here.
Yeah.
Hi, ma'am.
Hi, Vindhya. So Professor Maitra has joined? Professor
Yeah.
Maitra? Yeah, okay. I'm really sorry for this internet glitch. So I am happy that I could rejoin. So I welcome you all on this special lecture of Professor Uday Maitra. So this lecture is being organized by CRSI local chapter, Chandigarh, Amritsar. I hope Dr. Vijay is here.
Yeah, I'm here. Yes.
Yeah, hello Vijay. So he is the convener and I am the co-convener. So CRSI was established in 1999, and the objective of Chemical Research Society of India is to recognize promote and foster talent in chemistry and chemical sciences at different levels. So this year, Professor V.K. Singh is the president of the CRSI more recently, and various local chapters have been established. The main purpose is to reach at the root level. And on this context, the first week of November is being celebrated as CRSI Chemistry Week. And we are very lucky that to start with the celebrations, we have Professor Uday Maitra with us. Professor Uday Maitra is a renowned person in chemistry, and he is very closely associated with CRSI. He has served and is serving CRSI at different organizational level. So we are very lucky. We welcome Professor Uday Maitra to Amritsar, Chandigarh via online mode. And with these few words, I will like Mr. Jatin to introduce Professor Maitra. Jatin is a very bright student of chemistry department. He is BSc third year student, and he is very active in studies as well as in extracurricular activities. So Yatin, please introduce Professor Maitra.
Thank you so much for giving me this opportunity, ma'am. So first of all, good evening to everyone. I welcome all who have joined here in the celebration of CRSI National Week of Chemistry, organized by CRSI local chapter, Chandigarh, Amritsar. So our speaker for today is Professor Uday Maitra. As we know, he is a full professor at Department of Organic Chemistry, Indian Institute of Science, Bangalore. So brief introduction about him, you can also see on my screen, and I am very privileged to introduce him. He has done his BSc from Presidency College, Calcutta, and MSc from IIT Kanpur. Sir received his MPhil and PhD from Columbia University in 1986, working with Professor Ronald Breslow, and his postdoctoral fellow from University of California at Berkeley with Professor Paul Berlet. He returned to India, and after a year at IIT Kanpur, he moved to the Department of Organic Chemistry, Indian Institute of Science, Bangalore in 1989. His research interests are in the chemistry of bile acids, hydrogels, metallohydrogels, and organogels, organic, inorganic hybrid materials, enzyme sensing, and also his group has recently developed a journal strategy for low-cost paper-based photoluminescent enzyme sensors. He's also greatly interested in chemistry education and is a regular participant in a variety of outreach programs for high school and undergraduate students. He has received a number of awards and honors, including the most privileged award, that is S.S. Bhatnagar Award in Chemical Sciences in 2001, and is an elected fellow of Indian Academy of Sciences and Indian National Science Academy. So it is an honor, sir, to have you, and we are really excited for today's session on drug discovery, because actually this is a very trending topic in these COVID-19 days. So not taking much time, I would like to invite Professor Maitra to start with the session. Thank you, sir. Please, sir.
Thank you. And thank you very much for the generous introduction. I would also like to begin by thanking Professor Vandana Bhalla and Professor Vijay Anand and other office bearers of the CRSI Chandigarh Amritsar chapter for putting together this event. And I'm aware that many other local chapters around the country are also engaged this week in various activities of the CRSI local chapter. So it's really a great way to celebrate the National Chemistry Week. It's also very nice that we can reach a larger number of participants through the online mode, although it doesn't quite give you the in-person experience, but still the reach out sometimes is in fact little deeper. Okay, so I have chosen a topic, drug discovery, today for today's discussion, because there are many myths associated with drug discovery, particularly as Yatin mentioned, in the COVID-19 situation, as you have been reading in the newspapers almost every single day. Let me share my slides first, and I will straight away go to the slides, and I hope all of you can see this. So this is what I would like to discuss. Drug discovery, is it really that easy? And I will have an answer to this question in my penultimate slide. But I will go through the details of what is involved in the drug discovery process. But let me start with a disclaimer. The disclaimer is that I have neither participated in drug design, nor have I been involved in any medicinal chemistry projects. So therefore, I am basically an outsider, and therefore, you should keep this in mind that I'm not an expert in drug design or drug discovery. Whatever I'm saying is based on my own understanding and reading. Let me, switch off my video, because that's not necessary for, you need to see the slides only. Okay, so just to give you a broad introduction to the background of drug discovery, what I'm showing here, of course, you can say that I don't see anything, right? And neither do I, because this summarizes the entire metabolic pathway in the human body, okay? And it's very complex, and it's got many areas, many pathways, many steps involved in each pathway. And even if I zoom into this area, I would still see a fairly complex, highly networked pathway of reactions which are catalyzed largely by enzymes. So normally, that is, under normal circumstances, whether it is you or me, all these reactions are happening with very fine precision and extremely well networked. But once in a while, things don't go wrong, don't go right. In that case, things go wrong, and even if there is a problem somewhere over here, it can affect a completely different location of the metabolic pathway, and that can sometimes cause diseases. And what I would like to discuss today in the second half of my presentation are some very well-known and well-researched drugs, which are known for many decades now. Although towards the end, I will touch upon some of the drugs which are coming out in the more recent times. But what I really want to do in the first half of my presentation is to take you through a sort of a very broad overview of what exactly is involved in the drug design or drug discovery process. The material for my presentation have been taken from different sources. One of the source that you all can buy, in fact, is this textbook by Silverman and Holliday called Organic Chemistry of Drug Design and Drug Action. This is available in the Indian market as well, Indian edition. It has got all these chapters, starting with introduction, lead discovery, modification, receptors, enzymes, and so on. For today's discussion, I will focus to a large extent on enzymes and enzyme inhibition and inactivation, which can lead to the development of drugs. I will not touch upon DNA or other types of drugs, or for that matter, drug delivery. The other resource for my presentation are these annual reviews of medicinal chemistry published by the Medicinal Chemistry Division of the American Chemical Society. The latest one is 2019. So I have taken some material out of this book. And every year, in the beginning of the year, the Chemical and Engineering News typically would have some sort of a summary article about the drugs that were discovered or marketed in the previous year. So the January 20 issue in particular of this year has the information on the drugs developed in 2019. And I will touch upon that again towards the end of my presentation. So these are some of the resources. If you search for drug design, you will also find on the web many sources of information, but you need to take a look at some of the original data to make sure that the data or the description that you are getting is really authentic. All right. This is a summary of about 1,355 drugs which were approved during 1981 to 2010, almost 3 decades. And this pie chart essentially shows you the distribution of these drugs into various types. V stands for vaccines, 6%. B stands for biologicals, things like antibodies. N are natural products which are used directly. ND are natural product derived molecules. S is completely synthetic materials. So if you look at synthetic plus nature derived molecules which are further modified by synthetic chemists, they comprise more than 50% of this total pie. So therefore, organic chemists play a very important role in the design and discovery of drugs. And I will touch upon that a little bit later as well. If you now think about drugs which have really made big news, these are sort of like blockbuster movies. You know, the young people in the audience, you would always like to see a movie which is typically would be called a blockbuster movie, which has earned certain hundreds of crores in a certain period of time. I don't know, you guys know better. But there are also blockbuster drugs. And these drugs are the ones which can sell $1 billion globally in a year. So that amount of money in Indian rupees is now 7,400 crores or so. So if a drug molecule sells 7,400 crores worth of it around the world in a year, then it will be called a blockbuster drug. And I'll start showing you some of the blockbuster drugs of 2017, for which I have a complete picture. And this ring indicates that Humira, which is in fact a biological drug, which sold close to $19 billion. Okay, these are all units of billions of dollars. So you have to multiply it by 7,400 to get the crores, figure in crores of Indian money. And you can see that the highest one is close to 19, the lowest one is 4.6. So these were the top drugs in 2017. What happens really is that, you know, that the life of a blockbuster drug is not very long, because it is stays as a blockbuster drug typically till the patent expires. Once the patent expires, then other companies can make the molecule by cheaper routes and sell it more inexpensive way. So clopidogrel, a heart disease, which is used for heart disease very frequently, it basically is declotting drug. It doesn't allow the blood to clot. This was a blockbuster drug till about 2011 or 12 when the patent expired. And since then, the annual sales have dropped, if I remember the numbers correct, below $1 billion annually. So Humira is a sort of a different type, because for biologicals, it is not always very easy to make a generic drug. And that's why it is still selling very well, despite the fact that its patent, I believe, is over. If you look at the all-time hits, like, you know, there are all-time hit movies, the drugs which have sold a lot over the last 10 or 15 years, number one is Lipitor, a cholesterol-lowering drug. And till last year, the total annual sales was $164 billion. The second one is Humira, $137 billion, and it is still selling very high. So this may, in fact, overtake number one in the next few years. The third one is also $104 billion. So these numbers are really staggering numbers in terms of the money that each drug molecule, small or big, has earned for this company. So every company, therefore, would like to have a drug which will sell very well. Now, there's another way to look at these numbers. And the numbers, what I have shown here are 2018 and 2019. The circular chart I showed earlier was 2017. This is 18 and 19. And these top 10 molecules are still selling very well. Again, you see that Humira is still over $19 billion. Followed by this molecule, which is 11.1 last year. 7.2 in the previous year. So these are all top selling drugs of 2018 and 2019. And if you would now like to see the companies which market these drugs and their total annual sales, these numbers are also very large for the top 10 companies. Excuse me. Roche, for example, sold $48 billion worth of drugs in 2019, 44.6 in 2018. Again, I don't want to read out these numbers, but the lowest one is 21, 25 in the last couple of years. And therefore, you can see that these companies are very, very rich companies selling fairly high volumes of drug molecules, not just one, but many over the last several years. Now, it turns out that even though we talk about drug discovery and drug design, but in general, drugs are not directly discovered. Okay, this may sound very strange to many of you because you are reading about drug discovery and so on and so forth. But but to be precise, what is generally discovered is a small molecule, often called a lead molecule, a lead compound, which has favorable biological and pharmacological activity, but it has unwanted activities such as toxicity, other activities, absorption problem, solubility problem, metabolism problem. Now, how a lead compound is arrived at, and that is the drug design process. So I will not talk about that today, but that's where, identification of a target, lead optimization, and many of these processes are involved. But if you do find a lead compound, then what medicinal chemists, which basically is a subset of synthetic organic chemists, but with a more focus, what they do is to modify this lead compound by synthesis, chemically modify the structure, make different substitution pattern, change the pharmacophore, so that the desired activity, that is the desired activity, is amplified and the unwanted activities are minimized. And therefore, it goes through several rounds of synthesis, evaluation, resynthesis, re-evaluation, and so on and so forth. Now, if you go through this cycle multiple times, then you get what could be called a drug candidate, which is worthy of extensive biological, pharmacological, toxicological, and animal studies. So until this stage, it is largely in vitro measurements, that is, measurements done in test tubes. But now you want to do biological, pharmacological, and animal studies. And if it clears this hurdle, then you get what could be called a clinical drug, a drug molecule which is ready for clinical trials in humans. Okay, you have also been reading a lot about human clinical trials for the vaccines, and I will touch upon this at the very end of the process, because the COVID-19 problem and its drug design, vaccine development, these are very new and has been going on for just a few months. Drug discovery is a time-consuming and very expensive process. To get one compound to the market, 10 compounds, again, I'm giving you very rough numbers. These numbers are not fixed. It's not always 10, it can be 5, it can be 15. But typically, up to 10 compounds go through human clinical trials, and as many as 10,000 compounds are evaluated in animal studies. It can take between 12 and 15 years, starting from the beginning. And the average cost used to be $1,500 million. Just to make it look big, I have put million and not 1.5 billion. But what I understand from based on last year's data is that the price has now gone up to $2,500 million. And you can calculate how much that is in Indian rupees. The clinical trials, that consists of 3 phases prior to drug approval. And therefore, the drug candidates, that is the 10 compounds that I mentioned earlier, which fail late in the process of the clinical trial, results in lots of loss or unrecovered financial loss for the company. And therefore, everybody wants to have a molecule which will become a blockbuster drug. Nobody's interested in developing a drug which will sell, let's say, even $100 million in a year, okay? Because the investment that you do over a period of 12 to 15 years is very, very large. Bit about clinical trials, but before that, I would like to highlight 2 important terms which are often used in medicinal chemistry. These are called PD or pharmacodynamics and PK or pharmacokinetics. In simple terms, pharmacokinetics or PK is what your body does to the drug once you take it orally or through injection. Whereas pharmacodynamics refers to what the drug does to your body. Now in pharmacokinetics, 4 processes happen when the drug is taken. It will get absorbed, distributed, metabolized, and eliminated eventually. So this process is summarized as A, D, M, and E. So pharmacokinetics essentially studies this absorption, distribution, metabolism, and elimination process, because that's what your body is doing to the drug once it is orally taken. It goes to the stomach and or in your bloodstream, and how it gets distributed, metabolized, and eliminated. And these are processes which are very important to understand what actually happens to the molecule. But the pharmacodynamics is what happens when the drug actually concentrates at the site of action. Does it elicit a pharmacological effect? Does it lead to a clinical response? And is the clinical response efficacy or toxicity or both? In that case, you have to improve efficacy and reduce toxicity. So these are the things that you would ultimately like to have once the drug is administered. And therefore, pharmacokinetics, pharmacodynamics are very, very important in the drug discovery process. And that's where during the clinical trials, a lot of time is spent. Now a little bit about the clinical trial. There are 3 phases. Again, in terms of this vaccine development for COVID-19, you are reading about it in the newspapers. Phase 1 trial, which typically takes between 6 to 18 months. This evaluates the safety, tolerability, pharmacokinetics properties, and pharmacological effects in healthy volunteers. It's not given to patients in phase 1. Once it clears this step, that means you find it safe, tolerable, no major side effects, and has favorable pharmacokinetics and pharmacological effects, then you start giving it to a few hundred diseased patients. And again, the effectiveness of the drug, again, side effects, other safety aspects, and the dosing, these are all evaluated during 1 to 3 year period. Whereas phase 3, which is really the one that takes much more time, is between 2 to 6 years, is a larger trial with several thousand patients in clinics and hospitals. Sometimes it is done across the continents if the disease is prevalent in many countries. And this is the one trial that establishes the efficacy of the drug and monitors adverse reactions from long-term use. And that's why you are reading things about the vaccine trial, that sometimes you need to wait for several months, if not years, to ensure that there are no long-term toxicity or long-term adverse side effects. And after all these things are done, the drug administration in the respective countries would have to approve the drug, and they have to go through the data taken during phase 1, phase 2, and phase 3 trials before it is approved for commercial use. And this can itself take several months to several years, depending upon how much data have been accumulated during the 3 phases. I would like to show you now some data about how the spending has happened in the last many years, 2 decades, 2+ decades, and how the number of drugs that came out in the market have fluctuated over the last 20 years. These are old data from 1996 to 2006. On the y-axis of the left panel, I have R&D spending by pharma companies in billions of dollars, and that essentially doubled in these 10 years. On the right-hand side, you see the number of new drug approvals, which was 50 plus in 1996, dropped below 20 in 2006. So the per drug cost went up nearly 4-fold for the successful drugs in these 10-year period. But in the last 10 to 15 years, things have started to look a little bit better since 2010. Till 2010, the new drug molecules which were approved every year hovered around 20 After that, it has shown signs of increase, except in 2016. This is the data for 2017, which was pretty good with about 45 new drugs approved. And in the last 2 or 3 years, 2018, in fact, was the best year so far, with 59 new drugs which were approved in 1 year. And this year also, it looks good, but it may not be as high as 59. So you can see that the drug discovery went through a slump till 2010, but after that it is showing signs of recovery, except for occasional years like 2013 and 2016. Okay, so to sum things up, what I already described, I would like to take an analogy of a funnel. In a funnel, typically you pour a lot of material, but sometimes what comes out at the end is very small. So the drug discovery process often is compared with a funnel, because at the top you are putting 5 to 10,000 compounds in the preclinical, before the preclinical, in animal studies and so on. In the preclinical trials, it drops to 250 compounds, because most others would have some problem in the AD-ME process, in the PD process, and so on. And then finally, 5 or 10 compounds go through phase 1, phase 2, and phase 3 clinical trials. Then there is FDA approval. And finally, if you are lucky, you get 1 compound out. And as I mentioned earlier, it can take $2.5 billion or more, and it can take 12 to 15 years. Now, these numbers and this time doesn't speak very highly about possibility of getting a drug out for COVID-19 soon. But I will touch upon it again, as I mentioned, towards the end of my presentation. Now let's get back to molecules. The small molecules, which comprise of the largest number of drugs in the market at the moment, they have been found by Christopher Lipinski in 1997. He made an observation that most medication drugs are relatively small and lipophilic. That means they're fat-soluble molecules. And what he also found is that they typically would have not more than 5 hydrogen bond donors, that is NH or OH bonds, not more than 10 hydrogen bond acceptors, molecular weight typically below 500, although there are more exceptions coming up now, and a partition coefficient log P, which is less than 5. Since 5 or multiples of 5 appear in these rules, that's why it is called Lipinski's rule of 5. But the reverse of the rule is not true. If you find a molecule which satisfies all these conditions, it may or may not be a drug, okay? Because being a drug requires many other factors which are not simply summarized by the presence of these parameters. Since I am going to focus only on enzyme inhibitors today, I thought I will give you a crash course on proteins and enzymes in 4 or 5 slides. And I hope you don't mind this. Many of you probably know the protein structure, functions, enzyme properties, and so on. But for those who do not, I will show a few slides. So proteins are typically made from the 20 common amino acids, where the R is different. And you start from a one-dimensional or the primary sequence of the protein, where A, B, C, D, E are the side chain of the corresponding amino acids. And from the one-dimensional molecular chain, ultimately you get building blocks. And predominantly there are 2 types of building blocks: the alpha helices shown on the left, beta sheets shown on the right. And many protein structures use a combination of alpha helices and beta sheets with various kinds of linkers, loops, and what have you, to put together the 3-dimensional structure of the protein or the enzyme. And the 3-dimensional structure of a protein looks very complex to organic chemists. Even for one of the smallest enzymes, which is shown here, this is called ribonuclease A, which is a very common enzyme. It has about 127 amino acids and a molecular weight of the order of 13 kilodaltons. But it still looks quite big to all of us. Now, enzymes have immense catalytic power. They are highly specific, and the catalytic activities of many enzymes in our body are very highly regulated because of the highly networked nature of the metabolic pathways. The catalytic activity enhancement in one part of the sequence of the metabolic pathway can affect another part. And therefore, these enzyme actions are very highly regulated. And the last point I have indicated here is how enzymes accelerate reactions. And this is again a statement which was made by Linus Pauling in the late 1940s. He suggested that the reason enzymes catalyze reaction is because they have specifically or selectively stabilized the transition states. Just to highlight the catalytic activity of an enzyme, which is very common, it's a zinc enzyme called carbonic anhydrase. It hydrates carbon dioxide, which is produced when the fuel is burnt, like glucose is burnt in our body. And this has to be solubilized in water and thrown out of the body. So this one molecule of carbonic anhydrase, in fact, hydrates 100,000 molecules of carbon dioxide per second. So in other words, every 10 microsecond, a carbon dioxide molecule is hydrated by one molecule of the enzyme. So it's quite fast. In the next few slides, I will show a little bit about the enzyme catalysis and inhibition. Again, using cartoons taken largely from Strayer's biochemistry textbook. You all have read about Emil Fischer's lock and key principle. So the lock is basically the enzyme whose active site I'm showing only in 2D, but it's really 3D, like the enzyme structure I showed before, where A, B, and C are the three binding pockets for the substrate, which is essentially a key having units which will fit into the respective binding pockets as shown in the enzyme substrate complex on the right. And then, of course, the enzyme has catalytic machinery which carries out a reaction on the bound substrate, and enzyme gets regenerated. But sometimes it is necessary for you to slow down the activity of an enzyme. And that can be done using a substrate look-alike, like this red molecule I've shown here, which looks like the substrate. So the enzyme gets fooled into binding it, but once it gets bound, there is no option of carrying out any catalysis on it because it's not a substrate. And therefore, this blocks the access of the green substrate to the enzyme active site. So enzyme cannot do anything on the substrate, particularly if the red molecule binds much more tightly compared to the green molecule. So this is called competitive inhibition because the inhibitor competes with the substrate for the same binding site. There are other types of inhibitions, and I'll show you just one more, which is called non-competitive inhibition, where the enzyme, which is shown in yellow here, it binds to another part of the substrate structure, but it changes the shape of the active site, and thereby, when the substrate can bind, the enzyme cannot carry out the catalysis. So in effect, this is also inhibiting the structure, the reaction catalyzed by the enzyme. If enzymes are so good, they catalyze all the important reactions in our body, why would you like to inhibit enzymes? There are two situations in which you would like to inhibit enzymes. But before I talk about it, let me also show you how you could distinguish between competitive inhibitors and non-competitive inhibitors. So this is done typically by carrying out enzyme kinetics. And I'm sure that many of you have read about this equation, which is called the Michaelis-Menten equation, in which you measure the speed of an enzymatic reaction as a function of substrate concentration and carry out and plot this thing in a double reciprocal manner, which is also called a Lineweaver-Burke plot. From this, you can find out the enzyme kinetic parameters such as the Michaelis constant and the Vmax, which is the maximum velocity from this point and this point. Now, if the reaction is now carried out in the presence of an inhibitor, and if the inhibitor is a competitive inhibitor, you will get a change in the slope, but it will pass through the same y-axis point, okay? It will still pass through 1 by Vmax. And I will not go into the details of why this is so. On the other hand, if you have a non-competitive inhibition, it cuts the y-axis at a different point, but it cuts the x-axis at the same point. So by doing enzyme kinetics, you can find out whether the inhibition is competitive and non-competitive. So the next question, again, what I already mentioned, is why do you'd like to inhibit enzymes? There are two situations. One is that the drugs can target enzymes which are present in pathogens but not in the host. For example, if you want to inhibit a bacterial enzyme or a viral enzyme. Or the other option is that certain disease states in our body can be caused by the product of an enzyme catalysis. If the product concentration increases, suppose it causes a problem, and we would like to reduce the concentration of the product and therefore cut down the enzyme's activity. So in both situations, you would like to inhibit the enzymes. And I'll show examples of both types. The first example I have taken, however, is not a drug, but it's a agrochemical which has been in the market for many years. And the background to this is the shikimate pathway or shikimic acid pathway. This is the pathway by which aromatic amino acids are synthesized in plants and also in bacteria. We cannot make aromatic amino acids in our bodies. We are entirely dependent on plant sources for the aromatic amino acids. And they are made by a fairly long sequence of reactions, enzymatic reactions, starting with glucose, ultimately leading to phenylalanine, tyrosine, and tryptophan. One of the key steps in this enzyme catalysis is the conversion of this molecule, shikimate 3-phosphate, with phosphoenol pyruvate. It's a coupling reaction in effect to produce, enol pyruvyl shikimate 3-phosphate catalyzed by this enzyme. Now this turns out to be the slowest step or the rate determining step in this entire pathway. So therefore, if you inhibit this step, you inhibit formation of phenylalanine, tyrosine, and tryptophan, and the plant would die. Now, why would you like to kill a plant? You obviously don't want to kill a crop, but alongside the crop, you have the weeds growing. So typically, if you find a way to inhibit this particular enzyme, you have a weed killer. And such a molecule called Roundup was developed by Monsanto, which is nothing but phosphonomethylglycine. This inhibits EPSP synthase, and this has been in the market. It's still available in India and many other countries as a weed killer. And this inhibits this step and can shut down the shikimic acid pathway. Okay, if you want to read a little bit more about what is called Roundup Ready crops, genetically engineered crops, this is one of the sites that gives you a description of this Roundup Ready crops. So let's move on to molecules which have been used as drugs, but they work by inhibiting enzymes. So since many of you are not practicing medicinal chemists or are not involved in drug design, I thought I should consider molecules which are very well known, which are already in the market, and you probably have seen or heard about patients taking some of these molecules. One of them is for reduction of hypertension or blood pressure. Now, blood pressure can be, increase in blood pressure can be caused by many pathways, and one of the pathways is the so-called angiotensin 1, angiotensin 2 pathway. Angiotensin 1 is a decapeptide shown on the top, and it is cut by an enzyme called angiotensin converting enzyme. The last two amino acids are cut here as indicated to produce this octapeptide called angiotensin 2. And this is one of the most potent blood pressure raising substances, causing increase in the blood pressure by two different pathways. Again, the details are not very important. But if the doctor determines that you have high blood pressure and it is caused by an enhanced activity of angiotensin converting enzyme, then clearly you would like to inhibit this enzyme to reduce the concentration of angiotensin 2 in your blood. And a couple of molecules which are shown on the left-hand side, captopril, enalapril, these were developed as ACE inhibitors in the 1980s, I believe. And these were very high selling drugs those days. These are still available in the market, but there are better and more effective inhibitors of both angiotensin converting enzyme and the enzyme which is involved in the previous step, which leads to the formation of this decapeptide by an enzyme catalyzed process involving renin. Okay, but these are molecules which have been known for many decades now. I will now focus on a molecule with which you all are familiar, and these are penicillins. So penicillins inhibit the biosynthesis of bacterial cell walls. So unlike the mammalian cells, the bacterial cells have cell walls to protect the unicellular organisms. And the cell wall essentially looks like a cloth, piece of cloth. And just to illustrate that, I've taken a picture of a small piece of cloth in a microscope. And what you see here are threads which are going vertically and horizontally in a sort of a network way, all right, interlocked manner, so that you have a piece of cloth. And what the bacterial cell wall is basically like a cloth which is wrapped in the form of a sphere, and it covers the entire bacterial machinery. Except that they don't use this kind of a stitching. The bacteria use threads, let's say vertical threads, which are derived from sugar units, and horizontal threads, which are derived from peptides. And let's expand on the circled area, and where you see these vertical sugar-derived threads and this cross-linking process involving a pentaglycine from one of the chains to a D-ala residue on another chain. So this is where this cross-linking happens between this glycine and this alanine. Okay. Now the enzyme which catalyzes this process obviously is a very important enzyme because it generates the bacterial cell wall. So if you inhibit this enzyme, you will inhibit the cell wall biosynthesis, and thereby you will slow down the growth of the bacteria. This is exactly what penicillin does. And it does by mimicking the shape of the D-ala residue. Here is a picture taken from Strayer's biochemistry book, and you see the a penicillin, which is a common one being amoxicillin these days. This is the beta-lactam unit, and here's the D-ala conformation. And you see that these two conformations are similar looking. So amoxicillin is a very common semi-synthetic penicillin, but then the bacteria, some bacteria have developed resistance against amoxicillin because of indiscriminate use to a large extent. So what this beta-lactamase does in the bacteria is that it cuts open this particular bond before the drug has a chance to inhibit the cell wall biosynthesizing enzyme. And clearly, these are the penicillin-resistant organisms, and sometimes they don't get affected by penicillins. But of course, the medicinal chemists are not lying idle. And the way to get around this problem is to administer amoxicillin along with a possible inhibitor which will inhibit beta-lactamase, this enzyme. So such a molecule, in fact, is also found in nature called clavulanate. It's typically used as a potassium salt. This, in fact, inhibits beta-lactamase, thereby increasing the life of amoxicillin so that it can inhibit the cell wall biosynthesizing enzyme. So this particular combination is sold in the market in many trade names, and one of them is called augmentin because clavulanate essentially augments the effect of amoxicillin for the penicillin-resistant bacteria. This is a structure of the cell wall biosynthesizing enzyme, and I've just thrown in here to illustrate that this is an enzyme which has not only alpha helices, but also a beta sheet structure shown in the center in the red. In the next slide, I will talk about a late onset neurodegenerative disease. And one such disease is called Parkinson's disease. And here's a picture from a textbook written by a British medical doctor towards the end of the 19th century, okay, 1890s. And we fast forward 100 years, and here you see the head of a Parkinsonian patient whose skull is drilled to insert what is often loosely called a brain pacemaker. It has been found that this uncontrolled shaking of the limbs is caused by a neurological disorder, and this so-called brain pacemaker can alleviate these symptoms to a large extent, to about 70%. This treatment came around the turn of the 21st century, between 20th and 21st century. This treatment is now available in some of the top hospitals in India, and it costs between 15 to 20 lakhs, plus of course the maintenance, because it is essentially like an electronic device inserted in the brain. Obviously, you know, none of us would like to be in this situation where the skull is drilled and something is put inside the head. But coming back to the medicinal chemistry of Parkinsonian disease, I will skip this PET images for now because I'm running a little bit behind time. Parkinson's disease is basically caused by a loss or lack of dopamine, a neurotransmitter in the brain. And dopamine is produced in two steps, from tyrosine by an enzyme which converts this OH to a second OH. Well, this OH to a dihydroxy compound called DOPA, dihydroxyphenylalanine. And this then undergoes a decarboxylation process leading to dopamine. Now, if dopamine is responsible, if lack of dopamine is responsible for Parkinson's disease, you may very well say that, well, give the patient dopamine and Parkinson's disease will be conquered. But unfortunately, it doesn't happen, because dopamine in blood, if you take it orally or inject in the body, it doesn't go to the brain because of the presence of what is called a blood-brain barrier. The brain doesn't need everything that is present in your blood. So it has a filtering mechanism by which it takes in only molecules that the brain needs. Okay, and dopamine by default is not such a molecule, it's too polar. So you may say, okay, well, I will then give the patient L-DOPA, because L-DOPA in the blood goes to L-DOPA in the brain, because this crosses the blood-brain barrier, and in the brain there is DOPA decarboxylase. So this will decarboxylate and produce dopamine. But then there's a problem here. The problem is that just like there is DOPA decarboxylase in the brain, there is DOPA decarboxylase in the blood. So the moment you give DOPA in the blood, part of it, or a good amount of it, gets converted to dopamine directly, and then you are back to square one. So to get DOPA into the brain, you really have to dose very highly. So what's the solution? The solution again is very straightforward. You need to find a molecule which will inhibit the blood DOPA decarboxylase, that is this enzyme, but one that will not cross the blood-brain barrier. Because if it goes and blocks the DOPA decarboxylase, then it will not form dopamine. So such a molecule, in fact, was developed by medicinal chemists, which is called carbidopa. And you see between DOPA and carbidopa, there is a small tinkering of the chemical structure here. And this has the right properties, as I mentioned. This inhibits the blood DOPA decarboxylase. It doesn't cross the blood-brain barrier. And along with L-DOPA, if you give carbidopa, it reduces DOPA intake by as much as 75%, which means that if in the absence of carbidopa, you are giving 400 milligrams to the patient, now you can give only 100 milligrams. And that's a big, not only big saving, but also it causes less work on your liver to dispose of the excess drug molecule that you're taking, whether it is DOPA or its metabolized product. The next slide, I will talk about another metabolic product which causes problems, but this time it affects only older men. And that's why I have listed the name of only men, very famous men, who all had this disease called gout, which causes very painful inflammation in the big toe. And this is because of the formation of uric acid. Uric acid is produced through the degradation of DNA, particularly the purine nucleic acid components, AMP and GMP, which eventually get converted to xanthine, and then by xanthine oxidase into uric acid. Hypoxanthine, of course, comes from adenine, but again, xanthine oxidase converts it to xanthine and then to uric acid. Uric acid has a low solubility in blood, and it can crystallize in the form of sodium urate in the joints, and such crystals can cause a lot of problem in the ball and socket joints in our body. So therefore, if somebody has high uric acid, you would like to inhibit xanthine oxidase. And such a molecule is in fact known for many decades now, but it is still being used. It's called allopurinol. And if you compare this molecule with hypoxanthine, you see that all you have done is to switch the position of the nitrogen and the CH here. In fact, in the body, what happens is the enzyme faithfully oxidizes it here. So allopurinol gets converted to alloxanthine. Okay, so this is in fact the pro-drug. This is the real drug which is formed in the body, but this inhibits the enzyme because the enzyme cannot oxidize this site. So this acts as an irreversible inhibitor of xanthine oxidase, lowers uric acid, and sometimes it can reduce the symptoms of joint pain. But I should also point out that despite being an old drug, which was marketed perhaps in the 60s, I don't quite remember, but there was a paper published in 1984, which shows that you can have severe renal, that is kidney toxicity, kidney problems because of allopurinol, the structure I showed over here. Therefore, if this happens, you have to be very careful about using allopurinol. And this, in fact, happened recently to one of my former students who was taking alloxanthine, and he had a severe problem. And then eventually it was found out that it is because of allopurinol toxicity. After switching to another inhibitor or another way to reduce uric acid, he was all right. I would like to finish just mentioning about cyclooxygenase inhibitors. These are drugs which many people take rather routinely, like aspirin, like ibuprofen, and more modern versions called linezolid, celecoxib. And I would also like to highlight one more molecule. But before that, cyclooxygenase, we have some indigenous cyclooxygenase which produces prostaglandins and which keeps our stomach linings in a smooth form. But when you have an infection or an injury, like a bacterial or a viral infection, or a cut, or a pain, or an accident, which causes inflammation, then a second version of cyclooxygenase called COX-2 gets induced. And if you would like to reduce the pain, reduce the fever, you really want to inhibit COX-2 and not COX-1 and COX-2 both. But some of the old painkillers inhibit both enzymes. And some of the modern ones like celecoxib and rofecoxib, which were marketed, I believe, about 20 years ago, maybe a few years more. These were the second, the sort of specific inhibitors of COX-2, and these things can be used in smaller quantities. But then it turned out that rofecoxib, also known as Vioxx, this was removed from the market in the early part of this century because of severe toxicity in certain patients. Therefore, one of the points I would like to make here is that even after you get a blockbuster drug, it can still may get withdrawn from the market because it simply had not, some of the side effects perhaps would be observed only after it is used by many more patients, hundreds of thousands of patients over a longer period of time, and it can still be withdrawn. Like Vioxx was withdrawn in around 2001 or 2, I believe. Okay, I would now like to finish in about 10 minutes. I hope that's okay with the organizers. And what I would like to do is to highlight, just flash about 10 slides to highlight some of the drugs which have been developed in the last 10, 15 years. I will not show every year's molecules, but 2006 is a special year. I would like to show you because two of the molecules I show here, these are just some representative molecules among all those which were discovered in 2006. These are not the only, 6 drugs. The darunavir was an anti-HIV molecule. It was developed by my friend Arun Ghosh, who is a professor in Purdue University, and he was responsible for the development of this molecule. And vorinostat, which is an anti-cancer molecule, was developed by my PhD advisor, the late Professor Ronald Breslow, before 2006. What I will do is that next 6 years I will skip, because there's no point just showing you some molecules. But something that I would like you to notice here is that the molecular structures of the new generation drugs are getting more and more complex, because the enzyme inhibition that I showed you so far, these were fairly straightforward approaches. Nowadays, more complex targets are being looked at, and therefore the molecules to have the specificity are getting more and more complex. So let's jump to 2012. In that year, you have seen 30 new molecular entities, but I would like to emphasize here that the United States still leads this new drug market. In 2012, 22, European Union 4, and Asian countries, just a few, but Japan and South Korea are now coming out with new drugs almost every year. As I said, they look at the structure of the molecules, and some of them look more complex than what you have seen before. 2013 was an important year for India, because saroglitazar, an anti-diabetic molecule, was reported from India, and this was again taken from the Medicinal Chemistry Review. This particular statement: it was the first drug to be discovered, developed, and approved wholly in India. But you see, Canada 2, Russia 1, India 1, Japan 5 in that year. US still leading the drug discovery process. 2016, again, I have chosen just a few molecules here, 5 molecules here. 2017, I have not listed the chemical structures, but what I would like to illustrate here is that this is a year in which the number of molecules was about 46, I believe. First time it crossed that kind of number. And these are some of the molecules that were reported in that year. I would like you to take a look at the molecule 4 on the left-hand side, and I will specifically spend 2 minutes on this molecule towards the end of my presentation. 2018, again, 50 drug molecules, close to 50 drug molecules. And one of the things that I would like to point out to you is that the treatment for some of these molecules are very, very expensive, even by the Western standards. And some representative molecules which were developed in 2017, at least at that time, the cost of wholesale acquisition cost given in these numbers are something like $14,000 per month, $13,000 per month, and so on and so forth. Because as the molecules are getting more and more complex, the synthesis becomes more difficult. The investment in developing the molecule is high, and therefore the pharma companies would like to recover part of the cost by selling it at a fairly high price. And some of these things are not affordable for third world countries and poorer countries, many Asian countries, and so on. So this is again a completely different discussion, and we will not get into that today. 2018, as I said, was the best year so far with 59 approvals. I don't have the structures. You can always look at it from some of the links of FDA. But I just want to point out that almost everything from A to Z were included in 2018, leaving aside just a few letters such as Q and so on. 2019 was also a good year, 48 approvals so far. And 2020, the data in FDA website is published till 22nd October. There were 42 approval. There is something that you may be hearing about repurposing of drugs. In fact, the 42nd molecule approved on 22nd October 2020 is remdesivir for certain kinds of treatments against COVID-19. So that was the latest approval in 2020. So far, and you can hope that there will be another few molecules coming up in the next couple of months. I mentioned earlier that I will speak specifically about a molecule shown that was marketed in 2017, and the background to that is shown by this somewhat unrelated, what you may think unrelated slide. What I'm showing here is a tobacco plant grown in 100% water, and water containing 30, 50, 60, and 70% D2O. And what it does is that the D2O essentially reduces the growth of the plant. And in a very simple way to explain this is because of the kinetic isotope effect. Replacement of a bond by one of the atoms of a bond by its heavier isotope typically would make the bond slightly stronger and would make it harder to break, thereby slowing down many reactions. Again, I'm summarizing kinetic isotope effect in one sentence, which is not scientifically correct perhaps, but this is a plot to show you that because of a lower 0-point energy of CD bond compared to CH, this has a higher bond energy and therefore it is harder to break. And it turns out that if you have a molecule, a drug molecule, which is metabolized by the oxidation of a CH bond, and if you replace that by a CD bond and you have a large kinetic isotope effect, what will then happen is that if you replace the CH drug by the CD drug or the deuterated drug, the metabolism in the ADME process, the metabolism will get slowed down. That basically means that the molecule will stay in your blood for a longer time, which is very good news, because then you have to take the medicine either at a longer intervals or you have to take less amount of the medicine. You can ignore the panel B and panel C. These are for other types of details, but panel A shows how the drug concentration gets affected as a function of time after you take the drug. Let's say this is for the deuterated, the proteated molecule, the molecule having a hydrogen, CH. You have taken lesser amount of the deuterated molecule, so its peak concentration is less, but it is still above the minimum inhibitory concentration, which should be somewhere here. But when the minimum inhibitory concentration goes below this point so soon for the proton, the CH molecule, for the CD molecule, because of a slower metabolism, it crosses the minimum inhibitory concentration after a much longer time, thereby making it much more longer acting and more effective. So using this principle, the molecule that I showed you earlier in 2017, this was the first deuterium containing drug molecule that was marketed. The CH3 compound was marketed many years ago, but this was the first deuterium containing compound that was marketed because it was found that this particular bond is oxidized. These bonds are oxidized by our liver to produce an OH, which then fragments into the OH and OH group here, thereby making the molecule more water soluble for excretion. And there is a substantial kinetic isotope effect. And that's why this company decided to put the CD3 groups here, and that was successful. I should point out that, random deuteration of a molecule will not help developing a drug, because even if I put CH3, replace these CH3s by CD3, it will not have any effect because those groups are probably not involved in the metabolism process. So you have to find out where it gets metabolized, how it gets metabolized, and if there's a kinetic isotope effect associated with it. So perhaps many more deuterium-containing drugs will come out in the market in the next 5 to 10 years. Some of them are already in different phases of clinical trial. Finally, what about COVID-19? I have not said anything about it because I know very little about this. But the point is that as of now, this is my summary slide, that no drug against this virus is currently available. What people are trying to do is to repurpose other antiviral drugs such as remdesivir, other drugs such as dexamethasone to alleviate certain symptoms that are caused by the virus. So it doesn't kill the virus as such. In terms of vaccines, it takes time. Some people say it can take 10 years for a normal vaccine to come out. Effective drug, as I mentioned, that typical time these days is 12 to 15 years. There are lots of regulatory approvals needed. may take longer than a vaccine. However, because of the combined effect of many countries, many pharmaceutical companies, there is a statement, this was made in a recent chemical and engineering news, that there is no reason why you could not speed up the drug development if you're really focused on it. And now during the pandemic, that's what is happening, that there's a lot of focus, and hopefully both the vaccine development and effective drug development will take lesser time than it has taken for other vaccines and drugs for other viral diseases. But then one has to be cautiously optimistic, because this statement is also part of this document, that clinical trials are notorious for not going well. So again, I'm going to repeat this. We have to be cautiously optimistic, but we cannot say that it will come in January 2021 or it will come in July 21. No one can really make a prediction, because we have to just wait and see how the development is taking place. And until a vaccine or a drug is completely safe, it should not be given to a larger population. That's what I believe in. So my talk is generally over, but then I started with a question: drug discovery, is it really that easy? So therefore, I've answered the question in one slide with these concluding statements. And I hope that you have seen during the last 1 hour or so that drug discovery is expensive, time-consuming, multidisciplinary, and high-risk process. It involves not just one scientist working in a small laboratory, but hundreds of scientists and thousands of patients. And you need a bit of luck to get to a drug, because success in drug discovery process is not always guaranteed. With this, I would like to close my presentation by thanking all of you for listening to my talk, and also to the organizers of the CRSI local chapter, Amritsar, Chandigarh local chapter, particularly Professor Vandana Bhalla, who has communicated with me during the last several weeks. And I'm so happy to be part of this celebration of the Chemistry Week. Thank you very much. Let me unshare the presentation.
Thank you, Professor Maitra, for very informative, very interesting talk. And you can see there is a lot of discussion which is already going on in the chat box.
Okay.
Yeah.
I'm here. I'm still here. It was not a robotic lecture.
No. It was very interesting.
It could have been. It could have been.
It was actually interesting, and the environment is very interesting. And there are a lot of questions, I think.
Okay.
So Yatin, yes, in the chat box.
Okay.
And I think there was some discussion also, and there was answers also. So I will pick up a few questions.
Or maybe you can ask them.
Yeah. Anyone who will like to ask the question? Students? So I could see Mr. Alok, Namika, you want to ask something? Tanya. So you are welcome. Please interact with Professor Maitra.
Yeah, I think there are some questions about reducing uric acid by diet restriction.
Yeah.
Yes, that is something that doctors would always suggest, but sometimes it may not be directly connected with the diet. It can be contributed partly by the diet and partly by your own metabolism. If the metabolism is too fast, if the degradation of the used DNA is way too rapid because of the increased activity of the xanthine oxidase, there is another pathway that I did not mention, which is called a hypoxanthine guanine phosphoribosyl transferase, HGPRTase activity, which essentially is an enzyme that reuses some of the DNA degradation product for reutilization. Okay, so sometimes it is not because of the it's not because of the xanthine oxidase activity that your uric acid level is high, but it is because the reutilization is slow by AGPRTAs. So both can contribute to high uric acid levels.
Okay. So, Mr. Alok, you want to ask something?
See, there are also, certain questions I see here in the chat box that, a healthy body does not require any drug. Okay, we have all the good bacteria, hormone and reactions. That doesn't happen with, 100 people. But 101st example, if you, for example, happen to consume food that has a lot of bacteria and your immune system fails to clear that, you obviously have a bacterial infection. During the winter time, it's approaching again. I advise all of you to wear masks, not only to take care of COVID-19, but also it will help you in getting normal respiratory diseases, because you might have noticed that the amount of other respiratory diseases has come down for two reasons. People are not going outdoor as much as they used to, but also because of wearing of masks. Because it prevents, our air is not particularly clean. Sometimes it may not be bacterial. It can be just an allergic reaction to, dust, particles, pollens, and things like that.
And there are some questions related to catalysis also.
Yeah. One again, Alok Behra has another question: why companies always market and sell only drugs? Why they are only in business, and not in humanity? Because, as I mentioned, drug development costs $2,500. So a private company, if you have to invest money, this is all business. If you have to invest money, then you have to recover the money. You cannot spend $2.5 billion and sell the medicine for a much cheaper price than it was produced. So somebody has to foot in the bill. In India, what happens is sometimes, if the patent expires, then of course you have a chance of reducing the cost of the drug, like how it happened for clopidogrel, like how it happened for Lipitor and so on. But sometimes the government needs to come in, and our government does come into the picture and subsidizes the cost of some of the more expensive medicines. And along with that, I also believe that for diseases for which the major pharma companies perhaps would never develop a drug because the market size may be small or the purchase power of the people can be small. So for certain kinds of diseases, perhaps the government needs to invest money in a focused manner for a target-driven drug discovery process. And I think I'm not an expert, as I said, but during the COVID-19, from what I hear from my friends who are involved in drug discovery process, that such a directive by the government involving multiple national and private institutions for a focused discovery of certain drugs which are important for us will be attempted. But as I mentioned in my last slide, that this requires very, optimum coordination among people with different expertise, biologists, chemists, pharmacologists, doctors, patients, and so on.
So there are a few more questions from Alok also. FDA approved some toxic drugs too before. Why we have to believe in FDA?
You have to believe in somebody. FDA is the US Federal Drug Administration. I don't think that, knowingly somebody will approve toxic drugs, because sometimes the toxicity appears only when it is used by many more people, by the masses over a longer period of time. And that's what happened for this drug molecule called Vioxx, rofecoxib that I mentioned. So sometimes the toxicity is not observed in the first phase. sometimes you have to, particularly for terminal diseases, you have to look at, the toxicity versus the chance of survival. If a patient is dying, even though the drug is not fully approved, sometimes let's say in phase 3 clinical trial, but not FDA approved, but there can be a special recommendation that the pharma companies can get to give it to a terminally ill patient, because a patient is going to die anyway. And in case of, even if with mild toxicity, the drug can help save the patient's life, why not?
There is another interesting question from Tanya. If we take medicine for longer duration, it alters the normal temperature and it affects the normal function. Then why to consume it? Green chemistry, organic chemistry can be option. Why don't we use plant material for treatment? Okay, plant materials, one of the difficulties is that we have many plant-based formulations and drugs, but their exact nature of action has not been scientifically shown. In particular, there may be many components in a plant product, plant extract, which particular molecule is responsible for the desired biological effect is not often known. And it is also wrong to assume that plant products are always safe. Nothing in the world is absolutely safe for you. Everything has some side effect or other. It's only a matter of the extent to which it is toxic. So therefore, yes, there are many unexplored plant products that might help us, but until we do a very systematic study, and the natural product chemistry was very strong in India until the 1970s, and then the interest started to sort of change in other directions. But there has been a revival of it, and in a more scientific way, people are trying to find out, the biological activities of various types of indigenous plant extracts, and let's hope that some indigenous drugs will come out of these. But it's not always a good idea to take a cocktail of many things when you do not really know what is, does it really work? If it is work, if it is in the folklore, and if your grandmother says that, if your mother says that, perhaps you can take it, but depending upon how severe your illness is. If it is just for taking haldi or adrak as a routine, a routine sort of karak chai tea or something, then it's okay. But if somebody has more serious issues, then I think one has to take professional help. That's what I would like to advise. Yeah. So, dear students, I again invite you, please interact with Professor Maitra directly. If you want to ask him, you can directly ask. So there are other questions also. There is one question: is deuterated drug safe for human body?
Yeah, see, if the molecule with CH has been found to be safe, the one with the CD is also found to be, will also be safer. Okay, because the amount of deuterium that you are taking in the drug, and eventually it will be lost as HDO. Our body does have a small amounts of deuterium. Okay, small amount. And taking 100 milligrams of a deuterated drug will not affect your HD levels. So it will be as safe, in fact, a little bit safer compared to the original drug, because you have to take lesser amounts of that drug. I had a table showing how much of the benazine and the deuteriobenazine were given to patients. But if I remember it right, it was less than half, which means that your liver will have to do less work on the metabolism process. If you take 100 milligrams versus 50 milligrams, the liver has to do less work on the metabolism process, and excretion will also become easier because you have to excrete lesser amounts of the drug.
Yeah. So Tanya, you want to say something?
Yeah, please feel free to unmute yourself and ask me directly. I'll be more than happy to take your question.
So there are more questions from Alok. How can I help if someone is on to drugs? Okay. So I think he's talking about addiction, drug addiction.
Illegal drugs. Okay.
Yes.
Yeah. So I should have clarified this in my disclaimer that my focus is only on legal drugs, and there are many other issues about how you can help somebody who is on drugs.
Yeah, this
Is
is
on quote
Punjab.
unquote drugs, but that's beyond my expertise. That's definitely beyond my expertise. There was another question from Tanya, is that brain surgery was successful in that time than now? So plants become more positive effects. I don't understand this.
She is actually sporting drugs from the plant. Previous version also, in old times, we know Ayurveda was more advanced. Tanya, if you are here, so please unmute yourself so that we want to see you also.
See, as far as, drugs that are, old times Ayurveda was more advanced. We have no documented evidence for some of these. Okay. So unless it has been scientifically shown that this plant produces this particular molecule which takes care of this particular drug, you can say I'm absolutely fine if you believe in such medicines and you take it. But the point is that as far as modern day science and drug discovery is concerned, you have to have only single molecule. And you should know, in fact, I should also point out, I didn't have time to talk about it. If the molecule has 2 enantiomers, one such example is, I have shown you is, many molecules were chiral, but a common example is ibuprofen. Okay. Ibuprofen, what you take, if you have a toothache or a headache, strong headache, you take a combination of paracetamol and ibuprofen. Ibuprofen is also sold separately with the trade name Brufen. One of the trade names is Brufen. It's a racemic mixture. Okay. Although the S form is the biologically active form that inhibits COX. The R form does not. But interestingly, what happens is that some amount of the R form gets metabolized to the S form. It undergoes, inversion of stereochemistry to the S form. And the R form does not cause any damage to you. Okay, does not cause any toxicity. And I'm sure that many of you have heard about this thalidomide story. Yeah. Again, talking about it would have taken more time. But thalidomide was one case where it was found that giving both enantiomers can be a problem because one of them may be biologically active, the other one can have a toxicity effect. And nowadays, if a drug molecule, newly developed drug molecule is chiral, you have to make sure that the other enantiomer does not cause any toxicity, and you have to know the metabolism of both enantiomers. The enantiomeric molecules may be treated by our liver, by our body in different ways, different rates. So you have to show not only ADME for and PD, that is pharmacodynamics, for the right enantiomer, but also for the wrong enantiomer. And typically all new chiral molecules have to be marketed only in the one enantio enriched form. So that has made life much more difficult, more regulatory approvals are needed. And that's what takes more time for marketing of drugs.
So there is one more comment from Shreya. So can we consider that the drug discovery is very much difficult for the scientist without funding?
Of course, that goes without saying. $2.5 billion is what it will take to sort of go from scratch to a marketed product. Okay, and again, as I said, that not all drug discovery processes become successful. You may hit a hurdle after phase 1 trial, after phase 2 trial, or even after phase 3 trial. Okay, and then the later it happens, the more money you have lost in the process. So unless there is money involved, money given, there is no way to develop a drug because you have to go through this entire process, which is expensive. So I cannot simply say that, I will discover a drug sitting in the corner of my laboratory. Okay, all I can do is to find a lead molecule, and then somebody else will have to develop it from there. Okay, so what you read in newspapers, particularly in newspapers without any scientific publication, are probably the discussion of only the lead molecules, which has shown some activity in a test tube. That's it. Okay. And usually for such announcements in the newspapers, there are no follow-up news. Okay.
Yeah. Yeah.
So, so I often jokingly say, this is not to be recorded, that even if 1% of the drug molecules that are announced in newspapers were true, India would have been a rich country.
That is true. That is true.
Excuse me, sir, there are some questions from YouTube as well. So if you have permission, I can discuss that.
Yeah, please.
Sure. So first question, that is by Pravat Kumar, saying that biosynthesis of penicillin and cell wall, is it different from original penicillin synthesis, and how it can happen?
Okay, I did not talk about the biosynthesis of penicillin. Penicillin is produced by a fungal mold. It was discovered by Alexander Fleming under very unusual circumstances that you know. So that is a fungal product, but then that is chemically modified to make it more selective, more effective, like amoxicillin, which is a semi-synthetic penicillin. There are many others, but this inhibits bacterial cell wall biosynthesis. Okay. So we did not talk anything about penicillin biosynthesis, but we talked about penicillin inhibiting the cell wall synthesizing machinery.
Okay, so I hope his question is answered. And the next is that why drug testing in case of animals give quick result, but not in human being, or why we are doing it on animals, not on human being?
Okay. So as I mentioned in one of the early slides, that first you do it in test tubes, then you get a molecule that is worthy of biological and animal studies, and only after it clears the animal studies you test it in human trials. Nowadays there are certain ways by which you can guess the toxicity even before doing the experiments on human beings. And sometimes, even though animal studies may show proper results, the human body responds differently, because you use certain kinds of diseases in the animals, essentially as human models. Okay, but the way a rat or a mouse or a dog or a monkey would respond can often be different. And I'm told, again, I don't know for sure, that sometimes the dog models work better for human beings than monkey models, although you tend to believe that monkeys are more closely related to us. So there are issues like this, and so what you see in a test tube can be completely misleading, and what may work very well in a test tube may not work at all in our body because of the pharmacokinetic problem. It can have absorption problem, distribution problem, metabolism problem, and elimination problem. Okay, so until you do the pharmacokinetics, you would not even know whether it is going to act as a drug, whether it is actually going to the site of action. And even if it goes to the site of action, whether it will elicit a clinical response or it will have toxicity. So test tube results are very, very preliminary, and it should not be publicized at that point saying that I have discovered a drug.
So thank you for this question. And the another question is, what are the basis of Lipinski rule of 5?
Okay, it's really not a kind of a rule or a law, so to speak, but Lipinski basically looked at, his rule rule of 5 came in 1997. So the number of drug molecules known at that time, there were less biological molecules at that time, in fact. They were largely small molecules. So he started looking at the molecules and their properties, and they found some generalization. So it is more of a generalization than a rule. But what you can say, for example, naphthalene will never be a drug. Okay, it doesn't have any hydrogen bond donor, no hydrogen bond acceptor. It's very nonpolar, because ultimately, if you take it orally, it has to go to your bloodstream. It has to be solubilized. It has to go to the site of action, and it has to bind to a biological molecule. For binding to a biological molecule, you need some binding sites like an H bond or OH bond to form hydrogen bond, or take a hydrogen bond, accept a hydrogen bond from an enzyme. So unless your molecule is equipped with these and its shape is complementary to the biological active site, it will never bind. So Lipinski's rule essentially, talks about the sort of broad structural features that have been observed in a large number of small molecule drugs. That's all. But as I said, that the reverse is not true. If you make a flat molecule with 5 hydrogen bond donors and 10 hydrogen bond acceptors and a log p-value less than 5, it may become a drug, but it may not become a drug. Chances are that it will not be a drug.
And I think there is one more question in chat box, and someone like me is encouraged to perform these types of things at bachelor's level, and he or she is asking that how can we work in drug synthesis at bachelor's level? Or my question will be also in this case, what are the basis, or what skills should we develop either theoretically or either experimentally to work if we wish to work in drug synthesis in future?
Okay, this is very sad that in our country, we do not have any master's program in medicinal chemistry. This is something, I and some of my colleagues in various institutions, we have been lamenting about it, in fact, that we don't have any master's program in medicinal chemistry. And this is quite different from some of the other countries where they do have very strong medicinal chemistry programs. We do have pharmacy colleges offering B Pharm and M Pharm, but that's not exactly the same as far as I understand, not exactly the same as a medicinal chemistry, hardcore medicinal chemistry program in which you have to learn, a lot more than simply, enzyme inhibition and things of that kind. At the bachelor's level, there is no scope for now. The only way to encourage you would be to suggest that you do an internship in a pharma company, if that is possible. After master's level, if you have good skill in synthetic chemistry, then you can join a pharma company, but your skills will be to make molecules. But to really understand medicinal chemistry, you have to learn much more, and perhaps you should do a PhD before you can take up an interesting job in a pharma company.
Yeah, and I think Professor Maitra has answered Rinky's question also. Her question was almost same, that she is doing MSc in chemistry, and she was asking for some suggestions so that she can pursue her career in medicinal chemistry. And I hope you have got the answer.
Yeah, I also saw her question, and kind of combined these two.
Yeah. So Yatin, 2 questions are for you. People are asking for the Google form for the feedback.
Yeah, I think we are not having any feedback formats. You can obviously you all will be. I actually very much appreciated. Some my batchmates are also appreciated. I think that question, that girl is Rippan from my class, and
Okay.
yeah, I think I am going to contact her after this lecture. That how can we plan something like this? Yeah.
And what about the feedback form? Do we have any? Is there any possibility?
We can have it. Yeah, sure, we can have it.
Yeah, Professor Dr. Monubulan, he is asking for the feedback form. Okay.
Sure.
So do you have any other question?
There is another question here from Ravneet Steiny. What is the most easiest way to find binding site computationally in case ligand is not attached on the available target? Any software is available for the same?
Okay. I may not be able to answer this thing very directly, but based on what I understand from my, again, colleagues who do medicinal chemistry, see, sitting in front of a computer, and sometimes people say that, this is computational drug design. But so far as my knowledge goes, only one molecule was fully developed through computational work. But where computers can help is only if you have the 3-dimensional structure of the target known. Okay. If you do not have the structure of the enzyme or the other molecule receptor known, then computation will not help. But when you are doing the lead optimization, that is, you have a lead molecule which you know binds to the enzyme active site, for example, and you have the structure of the enzyme known, then you can do a bit of docking to find out which molecules you should not make. Okay, you chemically modify your lead molecule. All right, so there are many possibilities of putting various kinds of functional groups and so on. So having a computational chemist at that point, provided the 3D structure of the target molecule is known, can help you eliminate certain molecules that you should not make. Sometimes it can help you in optimizing the lead because you make a molecule and dock it, and ideally you should not just dock it on the computer, but you should actually co-crystallize the inhibitor with the enzyme and get its real structure. And that's what the serious drug discovery companies do, and that's what adds to the cost. That is, if you have an enzyme inhibitor that you have made and you try to get, let's say, you have a choice between 2 or 3 molecules. What they normally do is to crystallize the enzyme with all such molecules, get the structure of the enzyme inhibitor complex, then look at the, how these things are bound. Sometimes and then try to rationalize it by using docking studies. That can help sometimes, but it doesn't help all the time.
So there is one more question popped up from Namika. Is there any drug which have been ineffective to racemization?
Okay, certain drugs can racemize rather easily. For example, thalidomide can racemize, even ibuprofen can racemize. It all depends upon the chemistry involved. If it is a quaternary center, for example, okay, having no active hydrogen, it cannot racemize. Incidentally, I thought I will ask you a question. In the biosynthesis of the cell wall, I showed the structure of D-alanine, D-ala. But you all know that nature uses only L amino acids. But the bacterial cell wall has D-alanine. So maybe I will ask the students to find out how is D-alanine produced by this bacteria. I should not be the only one answering questions.
And that is true. That is true. So any other question? Anyone who want to ask anything from Professor Maitra? So he is associated very closely with the chemical education programs. So he is very friendly and he always welcomes the questions. So dear students, if you want to ask, you can talk in Hindi also. He will understand.
I should be able to understand some of it, not Punjabi though. Hindi I should be able to understand to some extent. I've given a lecture in Hindi in Raipur.
Okay, great.
And if you're still feeling shy, then you can email me your question and I will try to. See, the point is, since I'm not an expert, sometimes I do not have a ready-made answer to that, but I can find answers from my other friends.
Yeah, that
so
is another option.
Maitra at iisc.ac.in is my email address.
Otherwise, you can get it from me. And I think students have enjoyed the lecture because there were so many queries and discussion was all the time. Discussion was in the chat box. So, okay. So Yatin has written it. Yatin has already written your email ID.
Okay.
Yeah.
Oh,
so
yes.
Now,
I could have written it too. Yeah. Okay.
Yeah.
Maitra at IISc, at the rate.
At the rate.
Yeah,
At the rate is missing. So there are some comments for you, Professor Maitra.
Some more comments.
Mainly thanks, appreciation.
Oh, I am happy to know that. And, if any of you would like to have this PPT itself, I don't know if you recorded it, Vandana?
We have recorded it. It will be on YouTube.
Okay.
And
okay.
we will send the link to all, and it will be there on the
Sure.
YouTube.
Sure. You know, I keep updating this particular file. It was created some years ago, but every year I update it based on what's new.
Yeah. And I want to share one thing that there was some minor problem in Professor Maitra's eye. So, but he went up with the lecture and he sent me a recorded lecture also. And I have that.
You can delete that because I said things differently today.
Okay. So now over to Vijay for his comment.
Okay, thank you. So now we have come to the end of this wonderful webinar. So I'm sure actually most of the participants, I think most of them are students. They have got some basic information about the drug discovery and medicinal chemistry, some basic aspects of that. So on behalf of CRSI local chapter, Amritsar, Chandigarh, so I would like to thank Professor Uday Maitra for accepting our request and spending time with us through this online platform. I know everybody is having a lot of problems these days due to these trying times, but still one good thing happened in this trying time, right? So we are scientifically more connected through this session, right? So that is really interesting actually. So we have more and more interactions, and I hope this will continue for some time, even if the COVID situation ends. So this will continue, I guess. So that would be great. And so this drug discovery, this topic is kind of we consider this as an advanced level topic because unfortunately this portion, this particular area of area is missing in normal curriculum, I would say, right? So right. This considered as a special course in many institutes, including IITs, IISc, and even in other universities as well. So this is a very interesting and really challenging topic, I would say, because being in the industry, so I also worked in industry for some time, so I know the importance and how difficult it is to come up with a lead molecule. As Professor Maitra said, it is not so easy. It's really difficult task to come up with a lead molecule. So in that aspect, actually, this research area is really important and interesting, especially for students who wants to go to industry, especially discovery-based companies. And I take this opportunity to thank Dr. Bindya Arora and a very enthusiastic chap, Mr. Yatin Batra, is there. So and for their help actually in organizing this event, this webinar. And I also thank all the participants. Almost over 100 participants, participated through Zoom and YouTube channel today. So I thank all of them. I hope actually, I've got very, some take-home message, and about this drug discovery and all. So finally, again, I would like to thank Professor Maitra for giving this wonderful and inspiring lecture. Thank you.
Can I?
Yes, sure.
Can I answer one question which I'm noticing now?
Yeah.
This is from Jolly Behel. And the question is, we know there is no medicine for cold, but still doctors suggest us medicine and we get cured. So why this is so? There's a saying that if you have a common cold, I'm not saying anything more complex than that. If you have a common cold and take medicine, it will get cured in a week. If you do not take the medicine, it will take 7 days.
Yeah.
7 days.
That is good.
Of course, if you have any complication, you have to take an antibiotic. But common cold is caused by virus, which replicates rather quickly, which changes very quickly. That's why there is no real, antiviral. Even flu shots which are taken in the Western countries, they don't work, against all types of cold viruses. So basically, you take, supportive medicines, supportive things. If you have a pain or a throat infection, you gargle. If you have pain, you take a painkiller. or if you have a fever, you reduce the fever with painkillers. If you have a viral infection, only viral infection. But if it gets complicated, if you get a problem in your upper respiratory tract, and if you get a secondary bacterial infection after 3-4 days, then you have to take proper medicines. Okay. I thought I should answer that question.
Yeah, very nice of you, Professor. So I think this is for me. So this is very strange that Ripon, you don't have my contact. Okay, I will give it. So I think it is time to say goodbye to Professor Maitra.
Thank you.
And it was a great session, and it was a long session also, almost 1 hour and 45 minutes.
Yeah, I noticed it only now.
Yeah, we are very thankful to Professor Maitra. Maybe a cup of tea is waiting for you.
No, I have to make it in my office. There is nothing. You know, I'm actually not in the garden.
I understand that. I understand that.
This was a picture of our campus I took some months ago, and I thought I will put that as a background. But behind me, you know, which you don't see here, I have a coffee machine, so I have to make some coffee myself.
Okay. So thanks, Professor Maitra.
Thank you. Thank
Hope to
you.
see you
Stay
again.
safe. Wear
Yeah.
mask if you are going out. Students, don't make any compromise, even though the numbers seem to be going down around the country. Don't make a compromise until we have 0 cases all over the country.
Right.
Okay,
Take
then
care
bye.
and bye-bye.
Bye.
So I'm
Bye,
logging off.
Tisha.
Yeah,
Bye.
I'm logging off.
Yes. Thank you.
Okay.
Okay,
Bye.
bye-bye.
Bye.
See you. Bye-bye.